In this video, we're actually going to talk about how translation happens. And basically, it has everything to do with the three types of RNA involving protein synthesis all working together. You're going to have the messenger RNA attaching itself to the ribosomal RNA and the transfer RNA doing the same, carrying with it the amino acid that's appropriate for the code that the messenger RNA is asking for. To understand protein synthesis, though, you have to understand the structure of a ribosome. Ribosomes, remember, are made in the nucleus in an organelle that's called the nucleolus. What the nucleolus does, it will have pieces of RNA and protein which work together to decode a specific special type of RNA message, which comes from a special type of RNA polymerase, and it will transfer the RNA message into the actual pieces of the ribosome, which are made of RNA. Thing is, though, as much as 60% of the ribosome is protein. About 40% of the ribosome is made of RNA. The RNA will have both structural and catalytic roles within the ribosome. It will help the whole process take place. It will kind of like act like an enzyme, in other words. It will help create the active site inside the ribosome. Ribosomes actually have two basic parts. They have the large subunit and a small subunit. And you see that represented in several ways in this picture here. Notice that the large subunit will attach itself on top of the messenger RNA, and it will contain the E, P, and A sites, which we call the exit, the protein or peptide bond site, and the A for a waiting site. And we'll talk about that during protein synthesis. And then you have on the bottom the thing that holds the messenger RNA in place, which is a small subunit. Basically, what actually will happen is that when the messenger RNA exits the nucleus, a small subunit will attach itself to it almost immediately as soon as it finds it. And then, when the appropriate start codon is read by a transfer RNA, a large subunit will actually attach itself to it as well, and then the messenger RNA will be surrounded by the ribosome, and the actual translation process will start. And that's kind of how it works. Now let's talk about the actual translation process. It will happen in three stages, just like transcription: initiation, elongation, and termination. And it will include several players, including the ribosomal RNA, which is part of the ribosome, which is also made of proteins, the transfer RNA, which is carrying amino acids, and several proteins or enzymes called transcription, elongation, and termination factors. You're also going to have the messenger RNA, which carries the message from the nucleus, and several energy molecules, including ATP and GTP. Let's start with initiation. During initiation, the small subunit of the ribosome will actually attach itself to the bottom of the messenger RNA message until the appropriate transfer RNA, which is carrying methanol, remember, that is the equivalent to the start codon AUG. The anticodon on the transfer RNA will be UAC, and a methanol will be attached to that and put that in place. As soon as that happens, with the use of energy, or in the form of GTP, which is available, by the way, on that G cap, and that's what that's for, the large subunit will then attach itself to the top of the whole complex, and you're going to have what's called the translation initiation complex, with methanol sitting on the P site, or the peptide bond site of the actual ribosome. And the E site and the A site will be empty. Now, this whole thing would actually require the help of initiation factors. And without getting into too much detail, the initiation factors are a group of different molecules which will facilitate the attachment of the small ribosomal unit to the actual messenger RNA. Other molecules will facilitate the attachment of the large ribosomal unit to the actual initiation complex. Others will actually help each other out in actually making sure that the transfer RNA connects to the initiation complex. And so all of these proteins or enzymes are going to be part of what actually maintains the structure and function of the initiation complex. Without these proteins, it would be impossible to do protein synthesis, and that's one way that you can actually regulate protein synthesis, by destroying the initiation factors. If you destroy the initiation factors, no protein synthesis is going to be taking place. You're also going to need GTP. In most cases, initiation requires that jumpstart energy that's present in the G cap of the messenger RNA. It is possible to do it without it, but it's a much more complicated process. You're also going to need the start codon. So the complex is going to be reading the messenger RNA until it finds that AUG. And remember that the AUG is not necessarily going to be at the beginning, because after the G-cap you have an untranslated region. Remember that. And so that all of those initiation factors are going to be working together to help locate the actual start codon within the messenger RNA, and then initiate the process. Then a different group of initiation factors will help the transfer RNA attach to that and to help the large subunit attached to that. Now, once you have the initiation process completed, you're ready for actually elongation. Doing elongation, what actually happens is that one by one, amino acids are strung together to create the peptide bond by adding a new amino acid to what's called the A site, or the awaiting site of the ribosome. So let's say you have an amino acid sitting in the P site. Then you put another amino acid which has attachment to the transfer RNA, which has the anticodon that corresponds to the next codon of the sequence of the messenger RNA. That's the only transfer RNA that's going to be allowed to sit there. Only if you have the proper anticodon are you going to be allowed to sit in the A site. And since you're going to be holding an amino acid with you, you're basically going to put two amino acids next to each other. And those are the amino acids that need to be next to each other in the protein. And then a special enzyme that's called peptidotransferase is going to help create the actual connection between those amino acids. And then several different enzymes, which are called elongation factors, will facilitate the attachment of the transfer RNA to the messenger RNA and the translocation of the transfer RNAs from those sites. Let's see how that actually happens. Let's start at 12 o'clock when you see the new thing happening there. You're going to have an amino acid sitting in the chain already. You're going to have a transfer RNA sitting in the P site. Then, elongation factors will help the transfer RNA that matches the next codon attached to the A site if it has the proper anticodon. And it will be carrying the next amino acid in the sequence. With the use of energy and the help of the elongation factors and of peptidotransferase, you're going to create a new peptide bond between the amino acids which are now sitting next to each other. Then, with the help of other elongation factors, you cut the cord between the transfer RNA that was sitting in the P site. And now the protein is hanging from the transfer RNA sitting in the A site. Then, with the help of energy and another set of elongation factors, you're going to translocate. In other words, the transfer RNA sitting in the P site will move to the E site, and the transfer RNA sitting on the A site will move to the P site, and the whole thing will shift once over, and you're going to be basically back where you were at the beginning, because that transfer RNA that was sitting in the E site will just detach from the ribosome and leave to get another amino acid to repeat the process if it's needed. That's kind of how elongation takes place. I cannot stress enough how important it is to watch an actual animation of this to understand how this is actually happening. And understand also that this happens in a split second. In this picture, you see a more complicated description of exactly the same thing we talked about before. It all has to do with codon recognition, peptidotransfer, or actual making a new peptide bond and transferring the chain from the transfer RNA in the P site to the A site, and then translocating the transfer RNAs into the different sites within the ribosome. The ribosome shifts in the messenger RNA, and it's now ready to restart the process. And all of this, as you see, involves several different enzymes which are called elongation factors. Energy also in the form of GTP. Then, when you finally get to termination, what happens there is that eventually you reach the point in the mRNA where you get a stop codon. There is no transfer RNA that understands that stop codon. And so you're never going to get a transfer RNA to attach itself to the A site at that point. When you get there, a special enzyme that's called a release factor will attach itself instead of a transfer RNA attaching there. And that will send a signal to the actual ribosome that it's time to uncoil up. The subunits will separate in a process that consumes even more GTP, and then the protein is separated from the transfer RNAs, from the ribosomes, and the messenger RNA are all separated from each other. And then the release factor is also available to repeat its process again in another protein synthesis process that repeats itself over and over again. And that's kind of how termination takes place, and at that point you have completion of the protein synthesis process. Now notice that it's actually possible to do this in a string of ribosomes. Basically, one ribosome is working at the beginning of the messenger RNA, while the other ribosome is working right after that, and another one right after that, and another one right after that. So you basically have an assembly line of several ribosomes all working on the same messenger RNA, creating many protein chains from the same messenger RNA in a row. And this is called a polyribosome system, and it will create, instead of only one protein many proteins from the same messenger RNA. And if the cell does this, it will make a lot more protein than usual. A cell, for example, that's producing insulin in the pancreas is going to have many of these polyribosome complexes because it has to make insulin very fast to respond to an increase in sugar in your blood. On the next video, we're going to talk about how proteins are changed after the protein synthesis process is completed. I'll see you guys then.